Hello everyone, and welcome to the video on the sodium-potassium pump. The sodium-potassium pump is an enzyme found in the plasma membrane of all animal cells. It's a soluble pump that pumps sodium out of cells while pumping potassium into cells, both against concentration gradients. Since it goes against the gradient, it is an active form of a pump, which means it uses energy from ATP and is important for cell physiology. An example of where the sodium-potassium pump would be applicable in the human body is nerve conduction. The sodium-potassium pump was discovered in the 1950s by Jens Christian Skole, who was a Danish scientist that won the Nobel Prize in 1997. It marked an important step in understanding how ions get into and out of cells, and is particularly significant in nerve cells, which depend upon the pump for responding to stimuli and transmitting impulses. The sodium pump is a membrane protein that uses energy in the form of adenosine triphosphate, ATP, to perform active transport of sodium ions out of the cell in exchange for potassium ions into the cell. For every one ATP molecule, 3 molecules of sodium ions are pumped into the extracellular fluid, and 2 molecules of potassium ions are pumped into the cell. Therefore, there's a net export of a single positive charge per pump cycle. All mammals have 4 different sodium pump subtypes, or known as isoforms, and each has unique properties and tissues for expression patterns. So how the sodium-potassium pump works is that the pump, after binding the ATP, binds 3 intracellular sodium ions. The ATP is then hydrolyzed, which is an important term to know, leading to the phosphorylation of the pump at a highly conserved aspartate residue and subsequent release of ADP. After the release of ADP, a conformational change in the pump exposes the sodium ions to the outside, and the phosphorylated form of the pump has a low affinity for these sodium ions, so they are released. The pump then binds 2 extracellular potassium ions. This causes the dephosphorylation of the pump, reverting back to its previous state that allows for the potassium ions to be transported into the cell. The unphosphorylated form of the pump has a higher affinity for sodium ions than potassium ions, so there's 2 bound potassium ions are released. ATP binds again, and the process repeats. ATP provides the energy for the process to occur. The sodium-potassium pump helps maintain many factors within the body, such as resting potential, effect transport, and it regulates cellular volume. It also functions as a signal transducer integrator to regulate different pathways, as well as regulating the intracellular calcium levels. Overall, for the sodium-potassium pump, in most animal cells, it is responsible for 1/5 of the cell's energy expenditure. And for neurons, the sodium-potassium pump can be responsible for up to 2/3 of the cell's energy expenditure. The pump is vital for all living animals to live and maintain certain balances of molecules within their body. If this was helpful, leave a like, comment with any additional questions, and don't forget to subscribe to see more biology videos.